brothers and sisters, excuse me, sisters and brothers, humans are interacting, women and men, very complicated. Cells are interacting with each other, sometimes over proteins, very complicated. It's all about communication. There is not a single problem with a single solution, no. But by the way, what is intelligence? It's the eye of the observer.
Yes.
Hi.
I.
Yes, you are the eye of the observer.
Yes.
But watch out, we have the LSTM.
Uh-oh.
And we have new predictive modeling processes with machine learning.
Wow. How many rules?
Only 500,000.
Go on. That's not enough.
Okay, but look at the molecules. Look at these wonderful monitors. We have colors. We have structure, blue, green, and we put it together in this color and taste and everything. What you want, we can put all together.
Wow.
You can even go deeper in the structure, behaviors, and
Wow.
we have so many data.
But only 500,000 rules.
Yeah. But look at that, and a new thing is coming up. Steerable 3D convolutations.
Wow.
You can rotate everything, the 3D model. Wow.
Wow.
More general and so elegant.
Yes.
Okay, sometimes good correlation, sometimes not good correlation. Yeah, sometimes you want to change a little the atoms in a molecule. Yeah, sometimes.
Hey, that's my job.
Yeah, we know, but we have a new, it's all online. You can put it online. Colors and hidden. We have to find the good connections.
Me too.
Yes, this is a challenge.
Yes, for me too.
Let's go on in this wonderful afternoon while the dust is coming over the sea with 2 or 3 or 5 or 10 or 12 other speakers.
Okay, thanks, Martin, for this fun introduction. So let's start with session 2 with Sean Atkins, that I invite on stage. He has over 23 years of experience in pharmaceutical drug discovery. He graduated from the University of Aberdeen, receiving his master and PhD in clinical pharmacology in 92 and 96 respectively. He continued his research at Eli Lilly and Company Laboratories, Pfizer, and since 2001 he worked for a startup company, Concurrent Pharmaceuticals, as associate director, where he was responsible for developing computational models for MegaTox and targets of interest. He earned his doctor in science from the University of Aberdeen in 2005. And since 2006, Sean Atkins has consulted for several companies and carried out research on topics including, among others, pharmacophores for drug transporters, model for studying nuclear receptor ligand coevolution, and many other topics. He is currently founder and CEO of Collaborations Pharmaceuticals, which is focused on using machine learning approaches for rare and neglected disease drug discovery. So, thanks, Sean.
Thank you very much. It's a great pleasure to be here today, and I appreciate very much the invitation from the IPSA Foundation to be here. And I look forward to any questions that I don't cover in the next 24 minutes and 48 seconds. So I'm going to talk today about using machine learning, and as I go through the presentation, what I'd like to also impress upon you is how we've been able to build a company around using these technologies. I'm not going to stress so much on the algorithms, but I'd like to show what we've been able to do with them over the years, and perhaps also make the point that it's not necessarily the algorithms we should be worrying about, it's the other stuff that we don't really predict. And so I'm going to show you some of that, and that's really biology and not so much chemistry. So I'd like to move a little bit away from chemistry initially and start out with some biology. So just to give you a little bit of background about who we are as a company, we're actually a small drug discovery company that I founded in 2015. As of this week, we have 9 employees. We just hired 2 more. We are not funded by any VCs or angel investors. We're funded pretty much entirely through grants from the US government. These are small business grants, mostly grants that you would expect academics to be funded with, and then also funding from the DoD, Department of Defense. Our goal really is to try to bring novel treatments to the patient. And so some of the focus is not so much on coming up with new molecules, but repurposing old molecules and how we can use machine learning to do that. So I'm going to focus a bit on that initially. We do have some wet chemistry and biology expertise in-house, but most of our work is with collaborators. So this integrates very nicely with some of the previous discussions about how we need to be more collaborative. You'll see that a lot of the projects we work on are entirely collaborative. When we started out, we didn't even have a laboratory to do the research ourselves, so we're entirely dependent on the collaborators. And it wasn't until a couple of years ago that we moved into real laboratories with chemistry hood, as you can see here. We actually have 2 of those. We don't do chemistry ourselves, so we rely on buying molecules or working with expert collaborator chemists. So I have a bold vision, and really it's how can we industrialize rare disease drug discovery. There are over 7,000 rare diseases, and obviously many of these are not being worked on to any great extent. So how can we do that? And one of the approaches we think we can take is to come up with a method to scale drug discovery. What we tend to focus on in companies is either doing one thing really well or doing a few things really, really badly. What we'd like to do is to create a production line of molecules. And we think that we can leverage using artificial intelligence with real intelligence. And that, I think, is probably what should differentiate us from some of the other companies out there that are really focusing on just the artificial intelligence. You also need to have real intelligence to do some of this work. And I think that was emphasized heavily by Ed's 500,000 rules. So we've been able to create in a very short time a pipeline that works primarily on diseases that most pharma don't want to work on, diseases that are neglected or rare, ultra rare in some cases where they affect just a handful of patients. I'm not going to go through this pipeline to any great extent, but today I'm going to focus on our work on Ebola and Jacques disease, and then I'm also going to talk about a few rare diseases that are not really on this slide. But you can see that we can very quickly take molecules through to pretty much early clinical stage. Many of these are repurposed molecules. We've been able to get orphan drug designations for 3 of these from the FDA so far, and we've got a couple of other applications pending for Jacques disease as well. We also work on some diseases that impact much larger populations, like rhinovirus, the common cold, where there's been decades of experience but no drug so far approved. Obviously, that would impact billions of people. And norovirus, commonly known as the cruise ship virus. Huge unmet need, No one's really working on that. And so I think the new norm for drug discovery is this, very simple. Curate data, build models, generate predictions, perform experiments. That's really all we're doing. This is something that we've been trying to do for decades in the industry. I can go back to doing a postdoc in 1996, and I was essentially doing this. I just didn't know it at the time. I was curating very small numbers of molecules, building models, computational models, generating predictions, and then I was doing the experiments myself. And then I was feeding that information back around. At the time, I didn't think that this was any great shakes. Today, we're doing this now with massive data sets on the order of thousands, if not hundreds of thousands of compounds, in some cases maybe even larger. And the models we're building are not just one model, but thousands of models. And we're generating lots of predictions and ultimately trying to perform just one experiment. I think the first example I'm going to show you is the work we've been doing on Ebola. This started back in about 2015. And obviously, Ebola is back in the news because thousands of people are still dying in Africa. There are vaccines that are showing some promise, but there still isn't a small molecule drug that could be used either as a prophylactic or as a treatment to those with the virus. This is obviously impacting patients that are thousands of miles away from America. So why should we care about this? Well, obviously, we care about it when it comes into the news in Europe or in the US, when a patient comes back, or a potential patient. And then, obviously, the alarm goes off. We worry about it because it's in our neighborhood. So back in 2015, this was a big concern with the last outbreak. And so I looked to see what had been done. And a group had worked on doing a high-throughput screen in 2013 and published it. And I reached out to them and asked them if I could take the data set and build a machine learning model. And this small data set for less than 900 compounds allowed us to build a Bayesian machine learning model with commercial tools, screen a library of over 2,500 compounds, and we use that essentially to pick new molecules. So our approach is pretty straightforward. As was shown earlier, we have a list of molecules with properties. We generate simple molecular descriptors. These could be fingerprints or properties. We build a structure-activity relationship, and then we can also look at the feature contributions on the molecules. So with this initial data set that was published by this group in 2013, we were just trying to see if we could use that to find more active molecules. And we had a pretty high threshold of 50 micromolar. And so our goal was, could we find molecules that we could test pretty quickly? That was the easy part. We performed some other cross-validations with different machine learning methodologies. They essentially gave models, whether they were forests, trees, support vector machines, or Bayesian models. They were pretty similar in general in terms of the cross-validation. So no real big, huge differences there. And we also were trying to develop our own machine learning approaches at that time that we could use on a mobile phone. And the driver there was just to show that we could use the technologies in our pocket to do this kind of work, not just on our desktop. So we picked just 3 molecules from this 2,500 compound library that hadn't been tested before. And we were quite pleasantly surprised to show that they were all nanomolar compounds. They were able to inhibit the virus in cells. So they're able to kill the Ebola virus in cells in the test tube. Even more active than molecules that we had previously. And these are the molecules that were shown here. This is one of the few slides with structures for the non-chemists in the room. And you can see that they all have something in common. They have kind of a 3-ring core, which is a little bit different to the known antimalarial chloroquine, which only has 2 rings. But the bottom line is that these are all drugs. They're all already approved somewhere else in the world. So that should make the use of them and their approval for Ebola much easier. So this was really the start of our odyssey. Getting the in vitro data was easy. The rest of it is really years of work. So we did this in essentially 2015, and I'm going to show you data that's pretty fresh off the press over the next few slides. We had to build a team. Initially, we just had one group, Robert Davey and his team doing the in vitro work. We had to move into in vivo, and that's really where the costs mount massively. Imagine this work is done in a highly secure laboratory, a BSL-4 lab. There's only literally a handful of those around the world, and Robert had one of those. We had to then get money so that we could do in vivo work in animals, and a second lab, UTMB, University of Texas Medical Branch, got involved. And then we also have our chemistry and pharmacokinetics consultants. And more recently, we've even brought in the NIAID, National Institutes of Allergy and Infectious Disease, to help us really explore the mechanism of these compounds that we found. So we were able to get a grant from the NIH that was about $600,000 that we've used to generate data, which I'll show over the next few slides. So we progressed these in vitro hits, molecules that we were excited about. We've shown in mouse models that they protect the mice and have anywhere from about 70 to 100% survival of the mice. Mice infected with Ebola virus normally die within a week. And so now we're able to improve the survival of mice. And you're thinking, that's great, but they're mice. What about humans? Well, we haven't done humans yet, but we've done guinea pigs. And we've looked in mice at different dosing routes. We've used IP dosing, so this is the survival showing here whether infected and then treated 1 hour after dosing. We get 100% survival with a single dose of this drug, pyrenaridine, at 75 mg per kg. And we've shown after 2 hours and 24 hours of infection that we can also get about 80% survival in the mice as well with a single dose of this drug. That's right, a single drug dose. You take the drug once, it cures the mice. So normally drugs are given like daily or twice a day. This drug is just given once. It's got a really long half-life, and that's why it's an important antimalarial drug as well. We can also show that we knock down the levels of the Ebola virus to undetectable in the different assays that we can use, whether PCR or plaque assays. And so this was promising, and we submitted this to the FDA, and we were awarded the orphan drug designation in 2019. So as I mentioned, we've done mouse work. We've moved on to guinea pig work. And this summarizes our most recent work where we look at oral dosing. So the mouse work was done IP. This is doing oral dose. That's how the drug is given in the field for malaria. And interestingly, guinea pig is a very tough model. There are no published studies with guinea pig with small molecule drugs that have shown any success. People have been able to use monoclonal antibodies and other types of protein-type treatments, vaccines, but not small molecules. And what we've tried to do is do an analysis of all of these types of studies and use them as a control and show that pyrenaridine and favipiravir, another small molecule with a different mechanism, can protect the guinea pigs, but we don't see anywhere near the same level of survival. So we see about 40% survival. Again, the guinea pigs don't last very long when they're infected with Ebola, so we can greatly improve the survival. So that's where we are so far in our what we call our pathway to the clinic. We've done the Ebola guinea pig work. Because this is an approved drug shown here as part of a 2-drug combination used for malaria called Piramax, our next step is to try to use some of the registration information and see if we can get a pre-IND from the FDA. We're also trying to move forward and do a primate study with this drug. And then obviously we can use something called the animal rule and go straight into patients. So we can avoid doing clinical trial altogether in humans because of the unmet need here. So how does our dosing compare to the dose that we would expect? So a mouse dose can be scaled to a human, believe it or not. And there are well-published algorithms for doing that. And so this basically represents giving the mouse 75 mg per kg from our studies. This would represent giving the human just 2 pills, and they would have the same concentrations of the drug that protected the mouse. So at the moment, we're looking at the other drug that's in this combination called artesunate. We've tested that as well, and it has antiviral activity against Ebola of about 8 micromolar. And we're studying the mechanism of how these molecules actually work. And that's obviously quite important because then we can use that to design new molecules. We're also testing pyrenaridine against other viruses that are related, such as Marburg. The second example I'd like to briefly show you is Chagas disease. This was work that we did at about the same time as the Ebola work in 2015. This is a huge issue in South America. It affects millions of people infected with this. That is literally a parasite that's transmitted by this little bug here. It looks innocuous, but it basically poops on you, and this essentially spreads the parasite that gets to your heart and causes chronic congestive heart failure. So little do you know it, but decades later, when your heart starts to fail, this is the cause of it, in many cases in South America. So it's a huge problem. It's spreading into America, and I think some of the best estimates show that about 500,000 people are already infected in the US with this, specifically in the southern states. And there's only one drug that's approved in the US, and it's for children under the age of 12. And that's obviously no use if you're 50 or 60 dying from this particular disease. So we set out trying to use the same type of machine learning approach with our collaborator Jair Lage de Siqueiro Neto, who subsequently he's actually from Brazil, one of the largest countries that's impacted by this particular disease. And he's based at UC San Diego. And we published a paper in 2015 where we described our machine learning model using about 4,000 compounds that came from a published study at the Broad Institute in Boston. We built, again, a Bayesian model, screened about 7,500 compounds. We tested 99 compounds. We did a little bit, I guess, more compound testing for this. We had more money in the budget. We found about 17 hits. 5 of them were in the nanomole range. We tested all of those in mouse models that are shown here. And one of these compounds, again, was this pyranardine compound. Seemed to do miraculously well. It had about 85% success in treating the mice and destroying the parasites. So this molecule cropped up again at the same time. And so that was a very pleasant surprise. And subsequently, we applied to the NIH to get funding to do more in vivo work to see if we could use this molecule orally. And we've recently shown that we can dose this in mice up to 600 mg per kg and knock out the parasites to about the same level that we were seeing IP. So we have to increase the dose because of metabolism in the mouse. The standard that's used currently and approved for children is this benznidazole, which is an incredibly toxic drug to adult humans. And so what we're also interested in is lowering the dose of this and combining it with pyranardine. So we've now looked at the most recent study where we do a combination of benznidazole at 10 mg per kg instead of 100, and then pyranardine at 300 mg per kg, and we see about 86% inhibition of the parasite in the mouse in vivo, which is what we think is pretty acceptable to move forward into the next studies. I should say that these are all acute studies, which is very short-term infection of about a week. And now we're working on a very expensive study called a chronic study, which is basically treating the mice and then looking at them after 6 months. And this is the real acid test of whether this compound has any legs. And we're looking at a combination of pyranardine and benznidazole in this particular mouse model. And we're also going to do pathology, looking at heart inflammation and collagen deposition, as well as measuring other parameters. So this is a study that we have ongoing. It's funded by the NIH. We've already submitted orphan drug designation to the FDA for this compound. And so there's potential promise here that the same antimalarial can be used as an antiviral and potentially a novel antiparasitic. And if we're successful, we'll go straight into a clinical trial in Brazil. So all of these have a common point of reference in that we use Bayesian machine learning. And this has pushed us at the company to try to develop our own platform in-house for use, which we've called Assay Central, and take this approach of, well, if we can mine the public data out there and come up with models for Ebola and Chagas, we could do that for potentially thousands of other diseases. So we were able to get funding again from the NIH of about $1.7 million to develop our own software to do this, focusing not just on Bayesian, but other machine learning algorithms as well. And we've expanded this from Assay Central to focus on toxicity data sets, which we've called MegaTox. We got another grant recently from the NIH to develop this suite of software, and then also for repurposing molecules and also natural products, and we've called this MegaPredict. And this is using the thousands of models that we've built to come up with new ideas for new uses for molecules. And finally, our fourth product we've called Megatrends, which is using human uptake transporter data. And this is important for predicting drug-drug interactions. We've been applying the machine learning approaches to a number of the projects that we work on. One of them is coming up with new non-nucleoside reverse transcriptase inhibitors to go after HIV. We're aiming to find molecules that will be very effective and efficacious in the brain of humans. And so we've been modeling some of our relatively small data sets using our Bayesian machine learning methods. This is a data set of about 50 or so compounds with the TZM cells, looking at compounds that we've designed and tested. We can come up with pretty good ROC metrics. And then this is a published data set that's in the database called the NIAID, HIV and TB database. About 266 molecules. Again, we can get pretty good model statistics, ROCs of about 0.9 and 0.8 respectively. So we can use our models to do prospective design. We've been focusing over the last couple of years on doing comparisons with different machine learning algorithms. So obviously a lot of excitement about deep learning. And I'm also very cautious and try not to believe the hype. While this has been a very, obviously successful in other applications, we've seen a little glimpse of what it can do maybe with modeling some of the data sets that we have in the pharmaceutical industry. When we're looking at training, we seem to show that deep learning does pretty well, i.e. it trains very well. But what we've seen more recently looking at HIV data sets and others is that it doesn't really outperform. So here's deep learning in green. These are the rank normalized scores. We're not really outperforming other machine learning methods like Bayesian or support vector machines, whether doing cross-validation or looking at test sets. So the concern here is that maybe deep learning with these types of data sets isn't necessarily going to give us much of an edge, and we're probably good enough with the algorithms we already have at hand. And similarly, we can look in the environmental toxicology space, and we've looked at the estrogen receptor data sets, published on this most recently, and again shown that deep learning in the brown does not necessarily perform the best out of all the different algorithms that we have available. So I think there's still some caution. So in the last couple of minutes, I'd like to focus on why we need to do more machine learning in the rare disease space, because primarily what I'm seeing is that we have some very motivated families that are out there trying to save their children's lives, and many of them can raise lots of money very quickly. That in itself isn't a motivation for why we need to use machine learning, but it's just to point out that there is an increasing number of families that are motivated to get involved in drug discovery. And what does that tell you? They're going to go at some point towards the thing in their pocket, the phone. And what is that going to do for them in drug discovery? Not very much. So what could we do? Could we develop some of these approaches and make them accessible to these rare disease families? In some cases, there are already data sets out there. And so I point to this example for sialidosis. This is an ultra-rare lysosomal storage disease. It is an enzyme involved in the removal of sialic acid. It's also known as ML1. It's basically a hereditary disease. It impacts children. Most of them die before the age of 1. So there's not a lot of time to do anything once you've given the diagnosis. And what we've found is that there are some data sets out there where people have looked at the inhibition of this enzyme, sialidase, and we've been able to model those. The models aren't great, but we've been able to use them to come up with new molecules to test. And so far we've been able to find 2 molecules that have low micromolar activity, and this is some relatively recent data we've generated for one of these, showing a binding affinity of about 1 micromolar. And so the thinking is if we can find compounds that could stabilize the enzyme, that might give the children a little bit more hope and the families hope. And if we can do this for one disease, why couldn't we do it for 7,000? And so the final 1 minute, I'd just like to throw this at you. You're thinking, well, what is your IP strategy? You're doing a lot of repurposing. You're working on diseases where there's no patients. How are you going to make any money? Well, how are we going to make money? Should that even matter? We've so far been able to get 4 orphan drug designations. If we get those, we have 7 years exclusivity once we have an approved drug. All of the projects we work on have a priority voucher at the end. This is something that can be monetized, and each of these have been selling for hundreds of millions of dollars. So if we get a drug approved for Ebola, which we could do, we could get one of these vouchers, monetize it, and essentially have a massive return on investment. What is the future? In the last few seconds, I'd like to throw my prediction of the future. Everything we've spoken about so far is just the early stage. How do we use machine learning to find molecules? Well, there's so much more in drug discovery that we should be using machine learning for. It should be an end-to-end process. We've been looking at lead optimization, ADME, early stage drug discoveries, pretty much all of this. It's all been modeled, it's all been done over decades. What about clinical development? Can we use machine learning in clinical trial recruiting, design? And can we use it in post-approval? We can already use machine learning in cost effectiveness, fraud detection, adverse event prediction. So think about it as not just we can use machine learning to do drug discovery, but we can use it from cradle to grave. Essentially, I think the drug discovery scientists of the future will not be like me. They will have all of these technologies at their fingertips. They will have the ability to make these predictions, and then they'll have someone else that will do the in vitro work for them. And I think what we've tried to show is how that's possible. And it doesn't cost billions of dollars. It's literally a small company can work on lots of diseases simultaneously with a very small funding. With that, I'd like to acknowledge our small team, our many collaborators that I showed on some of the slides that worked on Ebola and Jacques disease and HIV, Vadim Makarov, I should highlight there. And finally, the NIH and the DoD who provided funding for us over the last couple of years. And thank you.
So I thank Sean Atkins for his talk. If you have questions, please keep them for later, because there will be a Q&A session at the end of the free talks, so that you will be able also to ask Atkins if you have these questions. Now we'll go for the second talk of Quentin Van Hellen. He will substitute Zavoronkov for illness problems. Quentin Van Hellen is the director of a virtual human project at In Silico Medicine, a bioinformatics and deep learning company based in Hong Kong, and focused on reinventing drug discovery and biomarker development. He graduated from University of Brussels with a master's degree in theoretical physics. He pursued a PhD in physics in Brussels, and after completing his PhD, moved to Canada to work at the Ottawa Hospital Research Institute. His research was focused on the modeling of the dynamics of signaling pathways and gene regulatory networks and their roles within stem cell fate decision. He then founded his consultancy company, In Silico Screen, focused on the analysis of signaling pathways, dynamics, and later acquired by In Silico Medicine in 2015. Please, Quentin.
Thank you for this nice introduction. Okay. So my talk of today will focus on describing the recent development we have done in building automatized pipeline based on deep learning for target identification and de novo molecular design. So I will go quickly to, okay, the first question is why are we doing this? So for those who are not familiar, this is like the general pipeline of when you want to develop a drug. So the different stage, so you have two different steps. You have the drug discovery itself and the drug development. The drug discovery itself is essentially a try when you want to cure disease. You would like to identify the right targets, and while you have a target of interest, you will try to find leads, so compounds which can actually bind or modulate this target in some way. So usually those process, once you have those leads, you try to optimize them so to make those properties much more better for a better result. And at some point you arrive at a stage where you need to prove that actually this drug is going to work. So you have different stages. The first time is preclinical stage, and if you are lucky, you will go through the next step, which is drug development, which include basically mainly 3 phases of clinical trials. So the big problem there is today is that most of the time, for a lot of hundreds of initial candidates you will have at the beginning, only a few of them will actually be able to go through these steps. And after like 10 years of development and hundreds of millions or maybe $1 billion spent, you will have at best one compound as a hand. And in most of the case, you have actually a failure at the phase 1 and phase 2. One of the reasons why we have this situation is that there is maybe a lack of optimization, complication when it comes to optimize and select the right compound we want to develop and until the very end. So the idea is to actually focus on the early stage of the drug discovery to optimize the way you select the initial compound and the way you can optimize them. So you can save time, and so it will be more cost effective and have probably lower attrition rate at the later stage, which are also the most expensive usually. So I think Silico Medicine, we are a deep learning based company, but we try actually to, initially we were based on target identification only, and then because we wanted to actually analyze and demonstrate that those targets were suitable, we started to develop de novo molecular engines to actually generate molecules which could actually bind those targets. So generator itself is based on what you know as generative adversarial networks and autoencoder and other kind of most recent deep learning architecture which has been developed in the recent years. And so to get you an idea how it looks like, this is like a general pipeline. So the first step is so the target identification itself, which rely also on a very good understanding of the biology behind the disease. So you need to understand what the disease is doing, the mechanistic mechanism of action of this disease, what part of the body is affected, and using all this data, transcriptomic, proteomic, and everything you can find, you can identify what could be the right target. Okay, or you can actually modulate this disease, stop this disease, or at least create some of the most deadly symptoms, for example. Once you have those targets, you want actually to generate novel molecules with very specific properties, so from a chemical point of view, structural point of view, so which make them suitable as to be used as a drug. And this is basically the core business of the company itself. And so, of course, as it was already said in the previous talk, you cannot go through this very complex process alone just by doing deep learning. Okay, so in addition, so if we want to go from this kind of algorithmic implementation to actually deliver drugs on the market, you need to collaborate with a lot of other organizations. So in our case, we have a lot of collaboration with universities all over the world, including in the US and Europe for very specific projects. We collaborate and have a lot of projects with big pharma companies, and also we have created a lot of startups. Each of them focus on very specific disease or biological questions that try to combine big data, AI, deep learning, and other more standard approaches in order to come with either suitable target and molecules. So now, so this is like a simplification of what we will try to do. So we want to develop, the idea behind developing this kind of platform is, of course, to optimize the way you can perform drug discovery, so improve this process, which will affect the way drug development itself can work and probably provide better results. And in addition, because we live in a world where now you have a lot of data which becomes more and more available, very easily available, so we will like, there is this kind of, I would say, positive feedback you can actually create. If you have access to how the drug is actually working on the patient, you can actually directly use this kind of information to actually optimize even further the way you actually design the drug. So you can go to what we will call, this idea behind personalized medicine. So we know that usually the response of patient to any kind of drug therapy may vary, not only because if you have a specific disease, you will have different pattern from different kind of patient cohort, and in addition, the way the disease will spread in the body, the way can vary. So you need to take that into account, and in order to take that into account, of course, you can. The idea is to combine all the data. So there is this idea of developing multimodal approaches. So rather than focusing on one type of data set, you try to aggregate all the data you can find, because each type of data, genomic, transcriptomic, proteomic, or imaging or even blood tests provide specific information on one side of what is happening. So it's just one part of this big picture. And so if you combine all of this, you can actually improve your understanding of the disease, improve your understanding of the mechanism of action, and come with a more specific answer to the problem for different kind of cohort of patients. So that's basically, I will say, conceptual approach, but basically which is, I think, quite interesting and which continue to be developed in the long term. So now, from a more, if we are talking about the perspective of developing algorithm for, generating molecules, this multimodal approach means that you have initially the knowledge of the structure of a molecule. You can look at this at different levels. So you have the primary structure, secondary structure, etc. And more complicated, you need also, and I think we had a very nice talk previously, explaining that this idea of having to model, or you have the drugs on your target are going to interact together. All this information can be encoded in different type of format. So typically as an input you will use either fingerprints, which are very commonly used in this field, smiles, or even chemical descriptor, and so on. So of course all this the way you encode the data, they are defined different ways. So you have the variability of some data, which can vary. So for example, 3D structure are probably providing a lot of information, very precise information, but they are not always available for all the protein you might be interested in. So it might not be very interesting to you to rely only on those kind of information. But you have also the problem of interpretability. So if you are building a model, especially deep learning model, you will have the features, the input, the output, and you want to be able to understand your output in terms of input. So obviously those characteristics will vary depending on the kind of information you are actually using. And this is actually currently one of the hot topic in deep learning. Sometimes the reason why people might be skeptical when using deep learning method for biological identification is that there are two big problems. You need to be sure, as I said, that you are able to interpret the result in a meaningful way. So not only in terms of very, I would say, abstract metrics, and also you have to be able to reproduce the result, because now we have so many different models which are built using different kind of metrics. So assess the performance, the reliability of all these models can be very, very complex. So this is also a problem you need to address. So obviously, in the case of reproducibility, it's obvious if you want to compare two models, see which one is performing the best, you need to know how to do it, and you need to specific metrics, common standard, I would say. And I think at the beginning, especially in the field of de novo molecular generation, basically everybody was coming with different kind of metrics, different kind of standard, and it's very, very hard to really understand what is the improvement behind all of these models. So because basically reproducibility, you want to compare this, you need basically to be able to compare all these models. And it means you need to compare models which, as you know, are very, very complex, because you have training parameters, you have the architecture, hyperparameters themselves. All of this you need to take into account to be able to actually perform meaningful comparison. And so the idea is that you need to develop some kind of platform which allow you to perform those comparisons. So one example is what we have done in Silico, is that you have a platform with usually most of the state-of-the-art baseline models, which are currently used in de novo molecular generation. You have a set of evaluation metrics everybody knows and agree with them, and also you have a standard data set you can use for pre-training, training, validation of your model. And so using this kind of automatized pipeline, which is part of the previous pipeline, you can actually have a better understanding what are the improvements when you're actually modifying a model by adding new feature or new module. So when it comes to generative chemistry, we have a lot of Western models which has been published. So most of them are, there's been like an explosion in publication with associated the release of the first generative adversarial network. Then you have autoencoder models and variational autoencoder. Those are the three main ones. And then the few one actually combining the concept of generative adversarial network with reinforcement learning. And so in the last 5 or 6 years, you had a lot of models which has been proven quite interesting and always based on different kind of input. So smiles, fingerprint are the most common, but you have also the idea of using molecular graphs, for example. And so when it comes for give you an idea for who's not maybe aware, but so for the gang RL model, so the idea is to combine this generative adversarial network framework. So you have on one side the generator try to actually generate the molecule, and then you have the discriminator will actually try to guess if the molecule it receives is actually a true one or a fake one generated by the model itself. And in addition, you had this reinforcement learning module to actually reward the discriminator to, according to how accurate its ability to distinguish between fake and real sample is. So that's one of the typical models which is commonly used today. And now when it comes to go a little bit further, so de novo molecular generation, you come with a set of molecules and you want to actually test them, so make them actually attractive for example, big pharma companies. Or so you have three things you need to focus on. Is that you need to prove that actually you are generating molecule you can synthesize, which basically show good properties. And for example, you need to show that they have good activity, and so you can actually is actually doing something. You need to provide molecules which are not already known, because reproducing what is already available is not always that useful, and probably come with different kind of molecules, so we can try different configuration, for example. So one experiment we have done recently is that we have released a result where basically the idea was to generate inhibitor of DDR1. So DDR1 is a famous molecule which is a collagen binding receptor, okay, which belongs to the RTK family. And the idea is that this DDR1, because of its position, is interacting with a lot of other receptors. And so as a result, it is involved in many biological processes. And as a result, and this is not really surprising, it is also involved with many kind of disease, for example, fibrosis, cancer, and so on. And the idea with this molecule is that you can try to inhibit or modulate its activity by either focusing on its interaction with the collagen, focusing on DDR expression itself, or kinase activity. So there are different ways to actually target this specific receptor, right? So in our case, actually, we focus on the idea so to inhibit this DDR1 kinase activity. And so why is it interesting is that currently it's not really obvious. So we know that DDR1 is involved with in many disease and processes, but the specificity of those interactions is also well known. And recently you had a lot of different kind of chemotypes which has been published for this molecule. And so the idea is that if we are able to generate a wider or bigger variety of DDR1 inhibitor, it might be useful to actually come with a one which might be suitable for different kind of indication. So in whole paper, the idea of the experiment was to demonstrate that we were able in a very short amount of time, so like 40 days, to come with a molecule, so starting from the initial target selection and initial set of data, and generating a lot of molecules using this model I will show after, spending some time like, actually as somebody has said previously, so the idea is you have to have the chemistry right, so in that case you need to go through a lot of selection and prioritization to make sure that your molecule meets the condition, and then select the molecule which were basically suitable for synthesization and synthesize the most interesting one and test that they were actually showing the expected kind of activity. So for the data set, I will not go to the details. The idea was to combine molecules from the zinc data set. I'll just show you previously, and probably you know this data. And then combine with also data about DDR1 kinase inhibitor, or the one which are actually common negative and positive set. And also we had a lot of some three-dimensional structure for the DDR1 inhibitor itself. So for the model itself, it's not actually a generative adversarial network in the standard sense. It's more like an advanced variational autoencoder with RL optimization, which was actually released like 2 years ago. So the idea was to use this model, which was already proven very useful, and to apply it to actually have a proven concept that you can push the generated molecule from the virtual world to the practical application, I would say. So that's just, and so as I said, the most step which takes the most of the time is actually the pre-training validation of the model itself. But you have another critical step, which is a prioritization. So how do you, because you have a model which generates like 30,000 unique valid structure in general, then you need to apply a lot of molecular descriptor tester, an additional condition to actually come with at the hand 40 molecules which actually are suitable for synthesis. And so in our case, basically we took the 6 most promising one, and they were actually synthesized and were proven to have, at least for the 2 first one, were proven to have quite significant inhibitory activity. So that was quite encouraging. And so the last one was also tested on a other model. So actually to test the pharmacokinetic of the model itself. And so all of these, the idea is that all of this took basically, 20 days to come with the list of compounds, and 40 days to come with the proof that actually this compound was actually working. And so this basically, of course, you have a lot of other things to do, because this is only a test on animal models. So if you want to actually go, for example, preclinical trial and then clinical trials, you have a lot of other additional experiments to perform. So this is basically a proof of concept. And this basically what distinguishes this kind of approach from many other works which have been done previously is that sometimes, because people making like de novo molecular generation, as we call it, are usually isolated. So they don't necessarily have this connection with the pharma company or chemistry company, which can actually synthesize a molecule. They don't necessarily have the biological understanding of the processes which are actually making this molecule useful. So they are maybe able to actually produce those molecules to generate those models, but they are not able to actually generate molecules which can be shown of interest. So here basically we see that if we are able to build like collaboration with people with entities with different expertise, you can actually go to this very early stage of target selection and take this compound, generate the compound of interest, and take it up to the actually animal testing. So this quite encouraging result, and I think this is actually the first of this kind which has been published. So in general, we can expect that in the following years you will have much more result of this kind, showing that even if at the beginning the novel molecular generation raised a lot of skepticism, I will say, because it was not done probably, in a different way compared to what the expectation of the field. Now you have, this field is gaining more maturation, and as a result, also more professionalism in the way the models are built, compared, tested, validated, and so you can start seeing that you can have concrete actually result. And in our case, actually, the DDR1 was not the first one generated molecule we were able actually to synthesize, and the first one actually is the JAK3, which is a common an inhibitor for JAK2, which is part of STAT2 activation and so on. And basically, we were also able to synthesize the inhibitor, which were actually generated, and show that it had also very good properties. So activity was actually significant. And as you can see, we have a lot of, several other successful projects on this kind. And so I think in the future, I think we will see many improvements in this very specific area. So thank you for your attention, and we'll take any questions.
So, thank you very much, Quentin, for your talk. Please stay around for the Q&A session. At the end of the session now, we should have A small break again before the last talk. The end.
Ah, okay. I was listening very carefully to the last wonderful, fast speaker. And I have probably the picture in my mind to show, I want to show you something about, you tell something to go really fast into a process. You want to have, a chemistry find, a drug find, a molecule find, and I want to share something too. I want to show you the sound finder. And the sound finder is something together with the microphone. So you want to go close to the machine learning, and this is my machine. And I learn to go really close to this machine. So I'm a machine learner sound system. And I want to show you, if I make a beatbox with bass without the microphone, it sounds dung, and this is really old-fashioned machine learning things. We need really the new AI and multiple layer learning IA, hopefully, but we saw the brown thing is not always the best. I go away with machine learning. That's really old-fashioned. I don't even know if everyone hears it. It's like and now the machine learns to bring the results, comes closer and closer, and you will see each year or step it comes closer, the sound will appear. Comes really deep, and it's a deep learning. $20 years ago. It starts. Self-driving cars. Deep learning. You have to go close to the deep learning. Deep learning. Maximum 2. Deep learning.
Oh, the Christ, it's the for all, it's gonna spell just the foundation for all. It's gonna foundation for all. It's gonna
Old-fashioned human thing, and now AI comes to the sound. And they go to the universe everywhere, but we stay here and we but we know since thousands of years, since the Chinese laughed about us, a new way. The fire.
pizza.
I tried to write something down. Last two wonderful content. Where they are? Okay. There are diseases, very public diseases, but there are also rare and neglected diseases. This is our thing. Our dream is, if we do it for one disease, we can do it for 7,000 diseases. It's always the same process. We are a small team, but we're 7,000. And we do it by scaling. Very easy. Doing the same process. For example, Ebola in Africa. In Africa doesn't it? Africa, but probably there's someone back from Africa to the US. Then we have to do something, yes. So we did machine learning very fast. We took 3 molecules out of 2,500 molecules. We found, but which one is the best? We needed a little bit of organization and collaboration, and a bit of money to put it together. Small team, and we took some mice. Yeah, take this here. And finally, we found out some pigs too. And then we found it. Puff! Suddenly, we had the right. This is the best. We took a wonderful packet, and now we have it. Yeah, medicine is ready for Ebola, for everywhere. And there is a star. Probably for us there is another star coming up. Deep learning. Yeah, it sounds great, deep learning. But we are not sure. You remember the brown? It's not always the best. The brown one, the algorithm is. We will see. But where there's another, we've dreamed. This is we don't need billions, we make it for less, but we will find 7,000 solutions. We don't need billions. In 48, there's another dream. In 48 days, finding the right molecules too, but it's more automated, the right medicine. How would we do that? Fully integrated, automated drug discovery AI pipeline. 48 days, a whole ecosystem around in silico. Oh, in silico. Where do you go? I go to in silico. In 48 days, I'm back to you. Yeah. We see us. Bye-bye. Testing. That molecule. Oh, fine. I'm out there. We want to individualize and personalize, different patient, it's clear, different results. It's not the other one, take care. But we found the right thing. So we found a multi-dimensional approach. Probably Probably I didn't get the whole message. But I think what is very important is zoom on GAN RLD models, RL models. That's right. It's very important, I think. And don't forget activity, novelty. The third I didn't get. I wanted to write down, but it's already so fast, man. But in 48 days, the whole process, this is a huge data. Honestly, wonderful fast. But so I have a refrain. Deep learning enables DDR1 inhibitor findings. 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 Inhibitor DDR, we should altogether inhibit the DDR1. Deep learning enables DDR1 inhibitor findings. And now please come a little bit together, shake hands with everyone and make like this, and probably with the fire lighting, and we sing the last time. Deep learning enables DDR1 inhibitor findings. Yeah, together. Deep learning enables DDR1 inhibitor findings. DDR1. Deep learning enables DDR1 inhibitor findings. Deep learning enables DDR1 inhibitor findings. And good luck. Thank you very much.
Okay, now we go for the last talk of the session with Alessandro Curioni, that I invite on stage. So Alessandro Curioni is Vice President of IBM Europe and Director of the IBM Research Lab in Zurich. On top of being responsible for IBM corporate research in Europe, he coordinates global research in IoT and security. He received his undergraduate degree in theoretical chemistry and his PhD from Scuola Normale Superiore di Pisa. In Italy, he started at IBM Research Zurich as a PhD student in 1993 before officially joining as a research staff member in 1998, where he had several research and manager roles, including being the founding manager of the Cognitive Computing and Computational Science Department. So, Alessandro, thanks a lot.
Thank you. Thank you very much for the introduction, and thank you for you to be still here after this performance. I don't know how one can speak after something like this, so whatever I'm doing probably will not be engaging. So, today we have heard at the beginning that deep learning and LSTM, if you wait a little bit of time, will solve all the problems, right? Then we have learned that indeed deep learning explainability of these black box models is a problem. We have to find a solution. We have also learned that more standard machine learning approach are eventually better or still better. And one of the major problems that we have with deep learning is data and the availability of data that are good enough in order to train good models. So what I will try to tell you, and I would like eventually that you remain after my talks, are three major things. One is that I do, from a research viewpoint, strongly believe that AI is going to transform the way we are doing R&D, not only in pharmaceutical industry, but in all the industries, that can help us not only to create models that allow us to get to our goals, but help us also to get the data that we do need, that we do have, but we don't know that we have, okay? Second thing that I want you to get is that AI is going with time, and especially deep learning, to transform the pharmaceutical industry, and will never be, as it is today, a question of having you know, a man that is doing all the dirty job to try to do the discovery and the synthesis and the test with what he has, but will be a man plus a machine, and will be judged in terms of results together, right? Today, you know, we have heard, and you will see also in my talks, we say we need to have the right models or the right references to understand how good a certain model and machine is doing for a certain task. We don't have these for humans, right? So putting the two things together, we will have really to look at the global output and outcome, and I do think that man and machine, with each of them, with their own capability, will be able to make a difference. So what is the problem? That is a little bit general, and then I will go deeper in pharmaceutical industry. What it is happening, has been happening in the past 20 and 30 years, is that mainly thanks to the first digital revolution, so digitization, right? We were able to automate things, to do things better. We were able to start and to create completely new sciences like computational sciences that will allow us to understand better the world. And automation allowed us also to increase the number of experiments that we can do. If you think already big chemical company today, they can do high throughput screening and screen many different reactions, millions of reactions every day, right? And we thought that this all together would allow us to discover faster and discover with a lower investment. So would allow us to have a higher return of investment, okay, in the full process. And this is not true and has not been true. And, you know, you can look, there are several economic papers that demonstrate this. You put in $1, you were putting in $1 50 years ago, you are getting outcome, now you put in $1, you get 5 times less, okay? And the major reason is that the problems that we are going to focus are very complex, but the way we are operating, right, in a linear way, we want to discover something, we have a target, we start to think with our knowledge the next step that we do, then we start to do simulation, we start to use databases and computers to create a space in which we search, and this expanded things more. We have high throughput. At the end, we go from something that was an idea goal to a huge amount of possibility, yes, for our discovery process, which of them, only few of them, will result in a new product, in this case, in a new drug, okay? And that is why, you know, the return of investment was going down. So, on the other hand, we do know, and I come back to the discussion that we had this morning, we do know that all these, for more than 20, 30 years, created an enormous amount of data and knowledge that we have up front, but that we cannot use it simply because we cannot assess. Think about a company, even a pharma company. The experience that is in the company is the experience of the person that is driving the discovery process, is the experience of the previous attempts that is written in reference documents. When the person disappears, or when the documents are written, even if they are stored in a database as a PDF, the knowledge goes away. So we are reinventing the same things or getting the same fundings multiple times, right? So if you start from that and you say, look, okay, I have this, but I have also other techniques that I call in the umbrella of artificial intelligence that would allow me to extract this knowledge, to create knowledge structure that then I can use, okay, to help me through different techniques, and I will show you some, to get the right decision, a decision or a movement that is orthogonal to the knowledge space that you have. It's not redoing the same things. We go from something that is very big to something that is small, and eventually we should be able to speed up the process simply because we are using all the knowledge that we have available. And today we are using a small one in space and a very small one in time. Okay? And that is a major problem. We have discussed today the data that pharma company has, the data that we have in databases. This data is structured data in relation database. It's a very small amount of the knowledge that we do have. Most of the knowledge that we do have is in this type of documents, is in paper, is in reference document, is internal documents, right? And we are able to take out only a very small, use a very small amount of this data and knowledge. Sometimes we do experiment, we do simulation, we do discovery process, and we go through the same failure simply because we don't know it. Yeah, we know only what is around us. And so that is why in IBM research, and especially in our teams in Zurich, we have spent a huge amount of time, and we're spending a huge amount of time, and now also with a much larger community, to try to develop what we call some cognitive discovery pipeline. That is something that goes from the bottom, right? Techniques that are using deep learning, by the way, are using AI to extract the knowledge from the document. And then we can put this knowledge in knowledge graph that are domain specific. We can use this knowledge graph to do deep search or as a source of data to train machine learning model, but with the right data and with the data with the right quality. And eventually going up, we would like to arrive in a situation in which eventually, I don't know when, we will have a full automated discovery. If you look in terms of where the technology is mature, I do think that we are now in the middle. The first two pieces we are able to do relatively efficiently. The second one we start to do, and we are moving up. Okay? To give even more argument why this is very important. Look, take, for example, a publication like Handbook of the Carbohydrates. What we put in these documents are table, images, and formulas, okay? Graphs. Most of the important information are in this type of document. The text, that is what most of us are already able to do relatively well with automatic processes, is only a glue for all the other data. If we are not able to extract all this knowledge from that document, we lose the important one. Yeah? We work on the text, we work on the structured database, it's very small and not important. I want to take it out. So I do need to have technology that is allowed to me to take it, to separate the things, classify the images, take out the data from the flowchart, from this table, understand the table, and then do the NLP analytics and put all these things together. Put all the things together, again, using advanced NLP. Example are here, where giving a simple set of rules, then they can help to learn the additional rules and convert all this information in knowledge graphs like the one that I do have here. This, if you look there, it's a knowledge graph for category of proteins that we have created, include 6 million of nodes. And that is something done fully from published papers and patents. Okay? And you see, in a graph like this, if you do things right, you'd have all your data, you know the relation of your data, and you can use, as I told you, eventually to do a deep semantic search on this data. Yeah, not a keyword search, but deep semantic search, okay? And eventually, you can use it to extract subset of data and feature that then eventually you use for the learning of your models, yeah? Any type of models, okay? You have the control of the data. You know how they are linked. You can test the quality. You can test the importance of the data. I take away a piece. I understand how the connection change. So you can do a lot of things if you have this structure. Okay? So let's move, and we are doing this in a very general context, exactly to try to do what I was telling you before. Let's look at pharma. So here, looking at the piece of the development is something that came out from a design thinking workshop that we have done with a few pharmaceutical companies, and is, you know, less, let's say, legacy in terms of the way it is done, simply because it was coming out of the discussion. But you see, there are different phases, you know, right? You have the discovery down there, structure generation, you have the synthesis that is part of the design, you have everything you have to do to make the drug with all the formulation, the test, and so on, and everything you have to do to test, yeah, preclinical, before going to the clinical trials. So these are all things and steps that eventually, with the help of data and with the help of the AI, we can not only automatize, but we can make it much better, yeah? And if you look, and that is something, you know, is my view, I may be wrong, but if you look at the full cycle and you look where already today AI is used or could be used, right? You see it there. So we have, of course, in the discovery, we have seen in using generative models and others. We can use it planning synthesis, and I will enter in detail there. We can use to decide best purification stuff, how we optimize the full reaction process. And of course, one of the things that many people have used is exactly the repositioning or the reformatting of the given compound, right? When you go to the test cycle. So what I'm going to discuss with you and show with you are two pieces that we have done in our lab, I do think, in the past 18 months. So it's not something very old. Yeah, it's not something that we have in a product or we plan to put in a product tomorrow. It's something that is advanced research in this field. One about the synthesis of these molecules, of new molecules, and then I will tell eventually, if I have time, something on some work that we are doing on generative models to propose, you know, new candidates, especially connected with personalized medicine. For the first, look, that I told you 18 months ago, we started to do this work. And what the team was came to me and asked, look, we do have this idea. There are people that are starting to use these deep learning methods to do a prediction of the outcome of chemical-organic reaction. And I discussing with the team, you know, I am a theoretical chemist. I have been a computational scientist for 20 years. I told them it will never work. Organic chemistry is too complex. There are too many factors. There is kinetics, there are conditions, there are solvents, there are too many things. I don't believe that a basic, only data-driven model with any experience added on top, right, will be able to do this well, okay? So the team started to do, and you know, the idea here was I have an organic molecule, few organic molecules, I put that together. I would like to see if I am able to predict the outcome of this chemical reaction. To predict the outcome, the product that come out, higher probability, the second, the third, and so on and so forth, okay? And eventually also if I change conditions, see if I am able to predict different type of outcomes. And there were people that have done using, let's say, standard deep learning and connecting the two things. It works relatively reasonably, but with a very relatively low accuracy and not fully reliable. What the team came out was, again, as other have tried to do in other contests, but not especially on this one, is to get to the consideration that a chemical formula has a smile type of a way, so a way to write the structure and the way the organic molecules is using a text, okay? And they thought, as Schmidt-Huber was saying, today we have LSTM-based models that are used to do translation between languages, that are, models that are relatively standard. They have what is called as an attention layer on top. That is something that allow to put more, let's say, more attention on the things that are important in a text, right? And here in this chemical text, they have used this framework. They have trained the model, and we were able, of course, to get a relatively good quality of results, but on top of it, having this attention layer, and this is what's called attention matrix, we can predict the product or the reaction, but we can also understand why that was the prediction, because these attention models on the two strings, right, tells you what was going to interact with in order to get to this final state. So we have not full explainability, but something along this direction, okay? And interesting things is that these models work not only on simple chemical reaction, but also very, very complex, like this Bromo-Suzuki coupling reaction. You have catalyst, palladium, several compounds, very complex things. It works very well, and it tells there, for example, the boron, right, that initiates the reaction, because activate one site is the initiator point of the reaction itself, and predict the right structure. To give you an idea on the accuracy of this stuff, look, this at the end is this new model. So accurate 92% if we have, let's say, cleaner setup with 350 Kelvin, right? 500 Kelvin goes to 90%. And you see, it is better than all the other previous models used, but it can give you an explanation why something is happening, okay? So once you start this, we have done an additional step, is to take all this framework we have put on the cloud, and we have made accessible to everybody for free, including the creation interface, the interaction with any database, so that people can test, try, and eventually bring back feedback to accelerate the development of this model. Then you can tell me, we also brought this to the American Chemical Society meeting and told people, look, this is a reaction, organic chemist, try to predict what is the outcome. The machine was better than the people, 3 times over 4, but with a major difference, 20 minutes for the person, 4 seconds for the machine to get to the answer, okay? And then you tell me, Alessandro, you told me all this, but how this feedback what we are saying before? Once you have this forward model, you can eventually use it to create models for retrosynthesis, only data-driven, okay? And retrosynthesis means you want to have a product, you know very well, so which are all the routes that you have to do in order to get there? Again, the machine can do it, and can create, you have a navigation in this big graph, can create not only one route, but multiple routes that then you can use depending on the constraints that you have. And you say, but yes, so this is important. is even more than important. Think about the transition from a drug that is going from, that is patent, and then expire the patent, you want to do a generic and you do the production in another place. With this stuff, you can find different routes, and you can also understand if the route that you have in the different condition can create problems for impurities and so on and so forth. And to give you an idea how it works and how fast it is, look, that is a molecule. I go there, I can tell, how many routes I want, I can tell the accuracy, I can tell how much computing power I do have, and so on and so forth. And I can tell, look, I would like at least to have this as an intermediate, right? And the machine automatically finds all the possible route and gives a priority using available product intermediate that are available on the market. So this is the time that he needs. Imagine, it's one molecule, he has to look at all the possible paths there, and then, it's a big graph. It takes around 40 seconds. And after this, luckily this is a movie, so we know that it's 40 seconds. But again, this it is something that depends on you. Yeah, it depends on how many. Yes, and you have for the final molecule all the steps. You go to an intermediate one, the second one, then you put together, and you can look at all the different sequences. Okay, this on the cloud with this accuracy. And I have 2 minutes. The same type of model that we have used there, that is using a deep neural network with an attention layer. We have recently used to predict here anti-cancer compound sensitive and sensitivities. And when we have done exactly the same, we have created the things, we have put on the web, is available on the cloud for everybody to test and give feedbacks. And what these things is able to do is able to, predict once you give a certain genetic profile for a cancer, the effectiveness of a given compound for that particular genetic profiles with all the mutation that it has. And having, these attention layers can try to help you to understand also why. And here is only one result of the latest studies is going to be published, in which looking at neuroblastoma case study, there you have the compound, if you use an average profile, there is the one looking at single cell lines, and you do see, so these is the IC50, right, so the activity, so that is a part of the effective compounds. There is the one unbiased, not optimized, and there is the one optimized. So you see that you can really come up and use these things also to go towards personalized medicine. And due to the fact that you can predict the activity, you can start to use these also for screening. So you can ask me, Alessandro, is how do you compare this with other methods? Because you could do these things in other ways. Is a very, very good question. My answer here is yes, we have to put together, we have to do comparison, we have to put together all these methods, but these type of techniques already now help the person that has to take the decision. It can be the physician, it can be the person that has to develop the drug, to move already in the right direction. Yeah? It is not fully accurate. It is not fully accurate also the organic chemistry prediction and the retrosynthesis one. But I tell you, is able already to do better than the men alone, and working together with the men, we should be able to get to much better results. So I finish here. I thought that I gave you an idea that these, AI general architecture are becoming general applicable. They can tackle problems that organic chemistry, come on, To become a very good organic chemist, you need to study university, you have to do a PhD, and eventually it's not enough, right? So we can do this fully data-driven in the machine. Data curation is the initial piece. If we don't do this, we cannot say deep learning is not working in this field. If I start from wrong data, okay, or data that are doesn't have the right features, the model that I have cannot be good. And we have to get this and understand this. And moving forward, of course, all these will change the way we are doing things. And I do think that, this interaction is the most important one. Thank you for your attention. Thank you.
So thanks a lot, Alessandro, for your very interesting talk. We are a little bit late, but let's maybe save just a few minutes for a couple of questions to the last three speakers. Yes?
I have a question for the last speaker. I like a lot the idea of open systems that can be given to the wide audience also to get the feedback. But aren't you scared about the quality of the feedback that you can get? Because this can spoil the system too.
Yes, but that is why I was saying you don't get this feedback directly in the system, and that is why I say that we still need to have an interaction with the person for now. But you can do also something more. It's not that you take the feedback and you put that in the original data and you rerun the model. You can take the feedback. I told you that we can create these knowledge graphs and knowledge structure that they have many connections, and you can try to understand if this feedback, right? So how this feedback modified the overall structure and the overall connection on this thing. It's not the ultimate answer, but it can help eventually to understand if it makes sense or not. Yeah, so.
Okay, thanks. Other questions? Okay, maybe if not, we'll stop here since we are a little bit late and we have to prepare the round table. So please stay here. At 6:30, we will have this round table around AI more in general, I would say. And
Andrea, I switch one moment to Italian. I just vorrei ringraziarvi di essere qui e anche comunicarvi che la città pare che sia tutta bloccata dal traffico, quindi io vi consiglio di rimanere con noi. Noi alle 6:30 cominciamo il convivium, quindi continuiamo ad affrontare il tema dell'intelligenza artificiale, dell'era digitale, ma da un altro punto di vista parliamo di un aspetto più etico e quindi se questo futuro poi sarà più umano o meno umano. Fondazione IPSA ha affrontato in questo anno l'intelligenza artificiale da diversi punti e il 21 maggio avevamo fatto un workshop a Milano al Museo della Scienza e della Tecnologia e avevamo parlato di umanità nell'era digitale. Oggi la parte farmaceutica biomedica che vi annuncio riaffronteremo perché come abbiamo visto c'è tanto ancora da fare, direi che siamo all'inizio e poi il convivio, quindi l'aspetto etico. Dopo il convivio, verso le 8, c'è una light dinner per chi resta con noi tutta la serata. Grazie e buon rientro.
We're just okay.